Okay, until now we have looked at Human Genome Project. What fields are emerging after the completion of Human Genome Project? Now, most new scientific disciplines do not arise entirely on their own. They are often formed by the amalgamation of knowledge from different areas of research. For example, stem cells, which is now considered an independent area, has its roots in cell biology, developmental physiology, developmental biology. But sometimes we also see that some old areas of research find some common ground. For example, if you go back since 1950s, like botany and zoology was entirely different areas. One is for plants and other is for animals. But now when you look at their cutting edge research, they are pretty much working at the forefront of cell biology, biochemistry and molecular biology. तो इस तरह आपको फील्ड्स का मर्जर होता नजर आता है और अब ज्यादा से ज्यादा जो रिसर्च हो रही है वो बेसिकली या तो इंटरफेसेस के ऊपर हो रही है या बाउंड्रीज के ऊपर हो रही है. नाउ फॉर ऑल द पीपल, and this is for you, for your future basically, that you would see कि कौन-कौन से ऐसे फील्ड्स हैं जिसमें आगे आप जाकर पीएचडी कर सकते हैं या फर्दर एमफिल कर सकते हैं. तो let me give you a bit of history and then come to the new fields. So Eric E. Kereli in 1999, who was an Hungarian agriculture engineer, coined the term biotechnology, and he wrote a book, Biotechnology of Meat, Fat and Milk Production in an Agriculture Large-Scale Form. But it took, from 1919, it took a lot of years that biotechnology became an independent field. So it takes a long time for a field to become an independent area of research, and as well as an area which is taught in all the academic institutes around the globe. So he defined biotechnology as all lines of work by which products are produced from raw materials with the aid of living things. But now we know, and we have seen previously as well, that this definition of biotechnology is old, and now pretty much whatever is done in the field of life sciences comes under biotechnology. But for a long time period, biotech process was considered only as traditional fermentation processes, where you take a raw material, you prep it up for fermentation known as upstream processing, and then you ferment it and ultimately do the downstream processing, basically clean the product and ultimately get a pure product in the end. But from 1919 till 1961, when Carl Goran Hedden basically changed the name of Journal of Microbiological and Biochemical Engineering and Technology to Biotechnology and Bioengineering, biotechnology was not considered a separate field. And when he changed the name of this particular journal to Biotechnology and Bioengineering, it ultimately started to be considered as a separate field. And then recombinant DNA technology came, and basically this whole theory emerged that we can basically clone foreign genes into other species. Particularly, it started with bacterial cell, and we started to get products, and biotechnology became an independent field. Similarly, after Human Genome Project, now genomics is one particular area. You can define it as a discipline in genetics that applies recombinant DNA, DNA sequencing methods, and bioinformatics to sequence, assemble, and analyze the function and structure of genomes. So genomics is one area, and then proteomics, which basically study the proteome. So rather than studying at the DNA level, we are discussing at the protein level. And study of the proteome, and what is a proteome? Proteome is the entire set of proteins produced or modified by an organism or system. This varies with the time and distinct requirements or stresses that a cell or organism undergoes. So basically, pretty much genomic content is usually the same, but because various genes can be turned on or turned off at various time points and in various cells, then you have to look at the protein content. And that basically, so when we are discussing about cancer patients, when we are discussing about various fields, we are looking at the upregulation and downregulation of various genes. And upregulation or downregulation of those genes basically give rise to variation in the protein content, which we try to map in the study of proteomics. But there are a lot of new fields coming out, like functional genomics, structural genomics, transcriptomics, metabolomics, epigenomics, and metagenomics. So basically, for example, transcriptomics is hugely important because that deals with that what genes have been switched off and what are switched on, and that basically ultimately looked at that how that particular cell line or organism is going to behave. And when we are talking about epigenomics, that whether any environmental factors or basically something which is not coded in the DNA basically affects the genome or not. So, but one area which is hugely interesting is systems biology. So up till now in biology, what we do, that if we find, for example, if I have found one drug target or one DNA or one receptor which I think is crucial in a physiological mechanism, I would try to isolate it. So मैं उसके पाथवे में जितनी प्रोटीन्स आ रही हैं, मैं आहिस्ता आहिस्ता उनको नॉकआउट करता जाऊंगा, और जब मैं उनको नॉकआउट करता जाऊंगा तो मैं कहूंगा अच्छा, अगर मैंने इस एक प्रोटीन को नॉकआउट कर दिया तो इस फंक्शन में ये चेंज आ गया, अगर मैंने इस प्रोटीन को नॉकआउट कर दिया तो इसमें ये चेंज आ गया, और अगर मैंने इसको नॉकआउट कर दिया और पूरा का पूरा पाथवे बंद हो गया तो इसका मतलब है ये बहुत की प्रोटीन है. बेसिकली दिस इज व्हाट वी कॉल रिडक्शनिस्ट अप्रोच, कि आप चीजों को आहिस्ता आहिस्ता माइनस करते जाएं और आहिस्ता आहिस्ता आपके सामने मैप क्लियर होता जाएगा कि बेसिकली असल के अंदर कौन कौन सी प्रोटीन्स थी जो पाथवे में इंपॉर्टेंट थी. बट सिस्टम्स बायोलॉजी इज basically looking it from the other angle. Rather than using this reductionist approach, they are going for integration. And it's not that reductionist approach is wrong or was wrong, because when it started तब आपको पूरे पाथवे की तमाम जितनी प्रोटीन्स होती थी वो पता भी नहीं होती थी. लेकिन पिछले 14-15 सालों में, especially after even prior to Human Genome Project, when people started to sequencing various chromosomes, they started to realize कि हमारे पास तो बहुत ज्यादा प्रोटीन्स हैं. और अब उसके बाद अल्टीमेटली नाउ वी हैव दिस होल मैश. सो इस बेसिकली आप प्रोटीन इस तस्वीर में आप बेसिकली इंटरेक्शंस देख रहे हैं कि अगर आप किसी को इंजेक्ट कर दें इन्फ्लुएंजा वैक्सीन के साथ, और इसमें जो रेड में आपको प्रोटीन्स नजर आ रही हैं वो बेसिकली अप रेगुलेटेड है और बाकी डाउन रेगुलेटेड है. So you can see, if you can look at these arrows, how they interact with each other. So ultimately, now we have the computing power to understand such various difficult images and pathways. And this has brought biology very close to computational world. And therefore, we need the aid of bioinformatics throughout in understanding all these mechanisms. And now a new field has emerged, which is known as synthetic biology. So various people define synthetic biology differently, but I would like to define it as an interdisciplinary branch of biology, combining disciplines such as biotechnology, evolutionary biology, molecular biology, systems biology, biophysics, computer engineering, and is in many ways related to genetic engineering. Easily you can say that designing and constructing biological devices, biological systems, and biological machines for useful purposes. If I may say कि synthetic biology basically एक engineer की approach है towards biology. So if you visualize this cell, and if I produce its cell mimic in terms of synthetic biology, then synthetic biologists would look at in terms of output, input, and basically more mechanistic way, that what are the parts which are giving various functions. Because until now, biology has been very abstract, but now we are giving it an engineering touch. There are various companies which are basically working in this area. So I urge you to go through this scheme which has been made by one of my friends, Justin Pahara, who runs a very successful company named Synbiota in Canada. And if you look at this graph, basically what he is telling you, that the companies involved with synthetic biology do scientific discovery, that they try to find drug targets, they do DNA sequencing, they do DNA synthesis, they do DNA engineering, and ultimately they are also involved in the experimentation to basically ultimately come up with more faster sequencing machines. And their end product producers who ultimately will produce the products are still traditional pharma companies like Roche, Lilly, Novo Nordisk, etc. So basically go through this image very carefully and look at all these companies. So basically this must have given you an idea that what sort of fields are emerging from in the aftermath of Human Genome Project. And these are the areas where you can carry your future research.